Chapter 20 is the study of genetics and human inheritance. As humans, we are 99.9% alike in our DNA sequencing, but that 1/10 of a percent, that 0.1, gives us our uniqueness and our individuality. The genetic information is carried on chromosomes in equal numbers in the egg and the sperm. The homologous pairs of chromosomes they're also referred to as homologs. There's 23 chromosomes that are received from one parent pair, with 23 chromosomes from the other parent. And each member of the homologous pair carries genes for the same trait. Genes are specific segments of DNA that are the functional units of inheritance. They code for a specific protein that will either play a structural or a functional role in the cell. The gene-determined protein can influence whether a certain trait or characteristic will develop. Thus, genes are considered the functional units of inheritance. Estimates are that there are about 20 to 25,000 genes that's distributed among the 23 pairs of chromosomes. A trait, also referred to as a characteristic, is produced by the actions of either one or more gene-directed proteins. Alleles is the term given for a different forms of a gene that produce different versions of the trait they determine. For example, the gene for freckles. One allele causes freckles to form, whereas if the other allele is present, then freckles do not form. This just is an image that shows a homologous pair of chromosomes, in other words, same chromosome that carry genes for the same traits. The gene is the specific segment of DNA that's considered the functional unit of inheritance. And a pair of alleles is the alternate form of a gene that's located at a specific site on a specific chromosome. One of these alleles will be inherited from the mother. The other allele will be inherited from the father. Homozygous is the term that defines individuals with two copies of the same allele. So these would be two chromosomes with the same gene, and in other words, homologous are called homologs. Heterozygous individuals are individuals with different alleles for a given gene. Dominant is the term used to describe when the effects of an allele can be detected regardless of the alternative allele, such as in a heterozygote. If you had a heterozygote that had two different forms, then if it were a dominant, it would override that recessive. Recessive is the term given when the effects of allele are masked in the heterozygote condition. Genotype is the term that's used to describe the alleles that are present, the genetic composition of the individual. In other words, whether they're homozygous dominant for a gene, or homozygous recessive for a gene, or heterozygotic for a gene. Pardon me. Phenotype is the observable physical trait of an individual. In other words, do they have freckles? Do they not have freckles? Do they have detached earlobes? Do they not have detached earlobes? But this is the observable physical traits of an individual, which also could be whether the individual had disorders such as sickle cell, anemia, or such. This just shows an image of an individual showing that freckles being a dominant allele, thus individual that was homozygous dominant or heterozygote for freckles would have the phenotype of freckles, whereas individual that was homozygous recessive, in other words, they do not have that dominant allele, would not have freckles. Widow's peak is another example of a dominant allele. So either a homozygous dominant or heterozygote would have the widow's peak, whereas an individual that was homozygous recessive would not possess the widow's peak. And this is just a breakdown of the table that you can find in your textbook, again showing that a homozygous dominant is represented by 2 capital letters, a heterozygote by a capital and a lowercase letter, and homozygous recessive by 2 lowercase letters. In this case, it designates F for freckles. The law of segregation is just a principle that states that during gametic formation, during formation of the egg and the sperm, the 2 alleles for each gene separate as the homologous chromosomes move toward opposite ends of the cell during meiosis. Each chromosome is inherited independent of other chromosomes, following the law of independent assortment. The law of independent assortment is the principle stating that different homologous chromosomes will travel randomly to different ends of the cell during the cell cycle. So members of each homologous pair of chromosomes segregate during meiosis 1, with each homolog going to a different daughter cell. Thus, an egg or sperm has only one member of each homologous pair. Each pair of homologous chromosomes line up at the midline independently of the other pairs. The orientation of maternal and paternal homologs relative to the poles of the cell is entirely random. Pairs of alleles for genes on different chromosomes thus segregate into gametes independently. And so this is just the image in your textbook showing the segregation that occurs during meiosis 1 and meiosis 2. Gregory Mendel is the individual attributed with beginning the rules of inheritance. He studied single genes inherited from parent to offspring. He did this in peas, looking at different pod size, flower color, so on. He first used a one-trait cross, which was considered a monohybrid cross, looking for one specific trait. Then he followed up with a dihybrid cross, which was basically looking at 2 traits. Did they have both yellow flowers and elongated pea size? A Punnett square is the matrix that we use to predict genetic makeup of offspring of individuals of particular genotypes. The rows represent the possible gametes of one parent, and the columns represent the possible gametes of the other parent. And so the boxes that we derive from this Punnett square represent the possible combinations of gametes that might be received from a cross between these individuals. So in this image it shows where we have a cross from the father, the sperm located at the top of the Punnett square. He was homozygous recessive because it's denoted by 2 lowercase. The sperm, the egg from the female, which is a homozygous dominant, notated by the 2 capital F's, would yield 4 heterozygote individuals, with the dominant allele for freckles being expressed in all. Thus, all the offspring would have freckles in this instance. A monohybrid cross is a cross in which both parents are heterozygote for one trait. In other words, in the example of freckles, they would be both capital L, little l. The genotypic ratio of offspring resulting from this monohybrid cross between two heterozygotes would be 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. So in the example of freckles, there would only be one out of the 4 possibilities that would not express freckles. This again is just an image of the sperm and the egg and the Punnett square denoted below. So basically, in the case between the cross between 2 heterozygotes, there's a 25% chance, or 1 in 4, that the child between 2 heterozygotes would not have freckles. There's a 75% chance, or 3 out of 4 chances, that offspring would express freckles in this instance between the cross from 2 heterozygotes. A dihybrid cross is a cross looking at 2 specific traits. So a dihybrid cross might be looking at if that individual had both freckles and a widow peak. So the dihybrid cross is a cross in which both parents are heterozygotes for 2 traits of interest. The phenotypic ratio of offspring that would be received from this would be the 9 to 3 to 3 to 1. So this takes us through meiosis 1 and meiosis 2, showing us the possible gametes that could be occurred. So thus, when we look at a dihybrid cross, instead in the Punnett square where we're looking at only a monohybrid cross and you had 4 squares, there would be 16 possible combinations, 4 to the 2nd power, which would be 4 times 4 when we look at a dihybrid cross. If we were looking at a trihybrid cross, it would be 4 to the 3rd power, which would be a 64 square combination. So in this instance of a dihybrid cross looking at freckles and widow's peak, there's a 9/16 chance that an individual that's 2 heterozygotes mating would have individuals that would be both freckled and both widow's peak. There would be a 3/16 chance that they would have freckles but not possess the widow's peak. 3/16 have no freckles but have widow's peak, and 1 out of 16 would have no freckles and no widow's peak. A pedigree is a chart that shows the genetic connections among individuals in a family, and the pedigree analysis is used by scientists to perform hereditary research. These are especially useful in following recessive alleles that are not visible in the heterozygote. Pedigrees are useful in determining an unknown genotype as well as predicting the chances one's offspring will display the trait. Family or medical records are used to fill in the pattern of the expressions of the trait in question for as many family members as possible. And this shows an example of a pedigree. Autosomal, when they look at this, or non-sex chromosome, dominant traits are typically seen in each generation. In other words, they don't skip a generation, such as the example that shows in A, whereas recessive traits, when they're looking at a pedigree, typically will skip a generation. The circles represent females. If it's not shaded in, it means they do not display that. They're not affected. The squares represent males. Again, not shaded in, unaffected, but shaded in, possess that trait. Genetic disorders are most often caused by recessive alleles. We know this to be the case because if not, you would see them both represented in homozygous dominant individuals as well as heterozygote individuals. A carrier is a term used to describe someone who displays the dominant phenotype but is heterozygote for that trait. So thus they can carry that recessive allele and can pass it to descendants, to their offspring. A dominant allele is one that often produces a functional protein. So the gene, the alleles for these, produces a functional protein. In other words, it functions, the structure, the function acts as it should. Whereas the recessive allele typically does not. Example of this is albinism. Albinism results from the ability to produce the brown pigment melanin that's in our skin, thus it's lacking. The ability to produce melanin depends on a specific enzyme. The dominant allele that results in the pigmentation produces a functional enzyme, whereas the recessive allele that results in albinism produces a non-functional enzyme. Complete dominance is a term used to describe a heterozygote that exhibits the trait associated with the dominant allele but not that of the recessive allele. Codominance is a term used to describe the effects when both alleles are apparent in the phenotype of a heterozygote. An example of this is in blood types such as AB. An individual with AB blood type would have protein products from both the A and the B alleles expressed on the surface of their red blood